And now we welcome on the stage Professor Alexandra Belayew, who paid a lot of attention the past 2 days, listening to all the research, and who will present the highlights of the research development. Thank you very much, Alexandra.
Thank you very much, Hisham. So I am feeling a little embarrassed to have to sit here, because usually when I teach, I'm running all the places, I'm jumping on the slides, but I cannot jump on 3 slides at the same time. Sorry about that. Okay, so the highlights means that I'm not going to talk about everything, but I take the opportunity to bring a reminder of what is the basis of FSHD. So, as big scientists do, I'm trying to do like them. I have to say my disclosure. So I might be biased when I speak about things done by Dyne Therapeutics, because they have the license to a patent we took in my university about a way to block DUX4. And I am also consultant for Merck, and this is very recent. And I think it's a very good idea for patients with FSHD that a big company like Merck is getting interested in their disease. Okay, so this is just reminding you what a cell looks like, with in gray in the middle the nucleus where the DNA is located. So DNA is part of the chromosomes. And this is a collection of recipes to make proteins. So you could consider that making a protein in a cell is similar to making a cookie. So in white on the slide is the connection with the cookie world, and in black is the connection to what scientists use as wording. And so the DNA is the recipe book. A gene is one recipe to make one protein. It can be copied as a messenger RNA. So this messenger goes out of the central library, which is the nucleus, and into the cell to bring the information to the kitchen, where the information is read by little workers in the cell to make the protein. Okay, so this is a quick reminder. And now, what about FSHD? You all know that your disease is caused by a genetic defect on chromosome 4. And so the horizontal black line is showing you the end, one end of chromosome 4, the area which is named 4q35. And there is something very strange. So you first have the large arrows are representing genes. So genes are only a part of your DNA, and there's plenty of other strange stuff. And what is very strange is close to the end, you have a set of repeated elements. So these are things that were long considered as useless. And by luck, a bright Chinese student in my laboratory named Hao Ding has discovered that within these repeated elements considered junk DNA, there was the DUX4 gene. So DUX4 stands for double homeobox on chromosome 4, so complex scientific name based on the structure of that gene. And how did we come from DUX4 to DUX? Okay, you all know Donald Duck. You know that Donald Duck sometimes can be very crazy, very mad. And from this idea, the name DUX, D-U-X, is pronounced like several ducks, like Donald. And we came to this crazy idea. You know, scientists are big children in some way. And so we found it funny to personalize this nasty protein that is causing you so much trouble by a nasty duck. Okay? So if we look closer at this set of repeated elements, I'm going to tell you what are the conditions to develop FSHD. So it's always said, even in the medical medium, that FSHD is such a complex disease, so difficult to understand. So I will show you that it can be really simplified. You need only 2 conditions to develop FSHD. The first condition is a genetic condition. You need a DUX4 gene. So where is this DUX4 gene? You have the set of repeated elements, so the repeated unit is named D4Z4. You probably have heard of that name previously. And when you make your genetic diagnosis, you are told how many D4Z4 units do you have. And so in each unit, you have the start of a DUX4 gene, you have the recipe to make the nasty duck, and that's it. So for a long time, it was a mystery. You cannot make a protein if you have a gene that is lacking an end. And so the end, very surprisingly, was only, was discovered by a bright Belgian graduate student in my lab, Eugénie Anceau. And the end of the last unit is outside of the repeated units. So the chromosome is longer than just the repeated units. And there's a region named PLAM, which is in pale gray on the right of the slide, that contains the end for a gene. And so the DUX4 gene is very strange. It is starting at the last unit, it's copying the recipe to make the duck, and it's ending at this signal named the poly-A signal, which is outside of the repeated units, which is something that nobody was expecting. And this is very important. This is defining what is named the 4qa allele, and people who have the 4qa allele have this signal of DUX4 gene end, which is absolutely necessary to make the messenger RNA with sufficient stability that it can go from the nucleus to the inside of the cell to get the protein made, and the protein is extremely toxic, and as I've shown here. Okay? So this is the first condition, the genetic condition. You need to have a full DUX4 gene with an end, so the 4qa allele. If you have a 4qb, you do not have this end, and you will not develop FSHD. Now, the second condition is epigenetic. So DNA is not naked in our cells, in our nuclei. It is associated with proteins, and this is making what is named chromatin. And chromatin can be of different types. So in order to have chromatin, you have to have DNA, proteins that bind to it, and DNA can have some modifications you've probably heard of in the field of FSHD, which is named methylation. So methylation is presented in my scheme by these little black dots that can come on some of the letters that constitute DNA, so the double helix on the right. So if you have methylation on DNA, certain proteins can bind to it and make a special chromatin. And so I will show you the two types of chromatin that are happening on the D4Z4 repeats, so on the DUX4 gene. If you are lucky, in healthy muscles, the gene, DUX4 gene, is off, not functioning. Why? Because chromatin is extremely compact. That's in the red square on the scheme. You see that the proteins are so tightly bound to DNA that DNA cannot be seen. You cannot see the DNA double helix anymore in that type of packaging. And this is happening when the DNA is carrying a lot of methyl groups. This is named hypermethylation. So hypermethylation is something good for FSHD. It's giving you a very tight, close chromatin, and the DUX4 gene cannot function. Unfortunately, for patients, in some way, the chromatin is open. It is in the green square there. So you see you have these little balls of proteins which are scattered on DNA, but you can still see the string of DNA. So the enzymes that come and copy DNA to make the messenger RNA have access to the gene, and the protein can be made. Okay? So in FSHD, chromatin is open to various levels, and the more it is open, the more DUX4 can be made. And this is associated with a small number of methyl groups, so DNA is said to be hypomethylated. Okay? So in these conditions, you can make the DUX4 protein. So how do you go from closed chromatin to open chromatin? Well, in FSHD1, it's just by having a small number of D4Z4 units. So for reasons which are complicated and that took a long time to scientists to understand, the fact that you have a small copy number of D4Z4 makes the chromatin open. Now, in FSHD2, you can have a higher number of units, up to 20 D4Z4 units. With 20 units, you don't get FSHD1. You need to have a second mutation. And this mutation is occurring on another gene, a gene like you probably heard this gene with a horrible name, which I will pronounce SMUSHT1. So this SMUSHT1 gene is involved in putting methyl group on DNA and in compacting chromatin. So SMUSH1 is a good guy, but when it is broken, it will not help chromatin to be tight. DNA will be less methylated, and chromatin will be more open, and this will allow for DUX4 expression. So SMUSH1 is the most frequent second mutation that you get in FSHD2, and there are other genes, one I mentioned there. So it has to be said that in order to develop FSHD2, you have to have two mutations. One is to have the DUX4 gene, like in FSHD1 with an and the second is a mutation on another gene that is affecting the chromatin structure. Okay, so I'm going to present data from few conferences that were in those two very interesting days for the researchers, yesterday and the day before, interspersed into the explanations that I give. So this importance of methylation in order to have a specific type of chromatin has been highlighted by Russell Butterfield from the University of Utah. And in Utah, there is the largest family in the world with FSHD. So you know the Mormon have a tendency to have one man and many women living with him, married with him, so that one single man can have many children. And the problem is that a patient with FSHD has moved to the Utah region from Europe in 1850, and he married many women there, had many children, and this family has been followed there for 9 generations. And so at each generation, you have 6, 7 people affected with FSHD. And this is fantastic matter for geneticists, of course. And so the data presented by Russell showed that these people have 5 D4Z4 units, and this is not changing across generations. So the copy number is the same over 9 generations, and the epigenetic condition is slightly different from one individual to the other, and the methylation level is really linked to the severity of disease. So if methylation is low, you have open chromatin, you produce more DUX4, and you have a higher severity. And the opposite is true. Okay, so that's very interesting because exactly within the same family, he could explain the variation that you have among patients. So the same genetic defect just changes in methylation, change the level of DUX4 expression and the severity. Okay, so I have to talk to you about another DUX4 gene. So besides the repeated elements that produce DUX4, to the left, you have one D4Z4 unit that in evolution has jumped and landed in the opposite orientation. And this is, and this is where you have the gene coding for DUX4c. So I had to give this explanation because you received this beautiful postcard. And I had to explain to you why DUX4c is the good guy and DUX4 is the bad guy, okay? So what are these guys doing? Okay, so DUX4c has normal functions in skeletal muscle, many functions for energy supply, for nucleus stability, plenty of things. I will not tell you all of them. And when DUX4 is in the same cell as DUX4c, then we have a big battle. So DUX4 is toxic to muscle just by itself. Why? Because it activates genes that are normally only active in embryo. So DUX4 is like an orchestra leader giving orders to various instruments to start playing. But in muscle, you don't have to have the violins that are necessary for early embryo. But with DUX4, you have a cacophony developing. Any instrument, all those genes are starting to play in your muscles. They have nothing to do there. And in addition, DUX4 is very similar to DUX4c, the good guy with the little heart. And DUX4 is a kind of gangster as compared to DUX4c. It would steal from DUX4c its target genes. So DUX4c activates some genes which are necessary in the muscle. But DUX4 will activate those genes way too much. And this is bad for the muscle. In addition, the two proteins, the two DUX, are very similar to each other, so they bind to other proteins in order to have activities in the cell. And DUX4, we hijack the proteins that are working with DUX4c and prevent DUX4c from doing its job. So that's why DUX4 does two things. It will kill muscle cells, and it will also interfere with muscle regeneration. And when you look at a representation of a muscle fiber, so the muscle is like a big rope made of small strings. So in the scheme, under the heading healthy muscle, you have what should happen normally in a muscle. So each of the red lines is a small blood vessel that is bringing oxygen and nutriments to the muscle. And the gray lines are muscle fibers, which are well aligned. You see that they are fat, they are filling the whole space. This is a good muscle. Now, if you look at the muscle from a patient with FSHD or from a mouse into which DUX4 has been put to be activated in skeletal muscle. So we have the same pattern. The mouse model of FSHD is the DUX4 mouse as compared to patient with FSHD. First thing, you have much fewer blood capillaries. You have only one red line on the scheme to the right under DUX4 muscle, okay? And then you have fibrosis. So fibrosis is deposits of collagens. It is something that happens when you have a scar on your skin. And you know that when you have a wound that becomes a scar, it is very rigid. So having fibrosis in your muscle is preventing it from working properly. You have inflammation. So you know when you have been bitten by a mosquito, you develop inflammation around the mosquito bite. It's becoming red, it's becoming hot, and this is happening also in your muscles. And then you have infiltration by immune cells. This is something that is starting to be more and more well studied in the field of FSHD and considered as extremely important. And finally, fat deposition. You know that what's happening with time is that your muscle is being replaced by fat. And this is all because of the fight between DUX4 and DUX4c. Now, this fat that I showed on this slide on a section of muscle, you can see it by whole body imaging. And these are fantastic pictures that are we have owing to the fantastic work of FSHD Global and especially Emma Weatherfield in collaboration with the company Springbok. So Springbok has artificial intelligence use to analyze data from whole body imaging and can distinguish normal muscle, which is presented here in red in these schemes, and fat accumulation, which is presented in yellow. And so you have here several individuals presented from the face and from the back. And to the left of the slide, you have patients which are less affected. You see they are more red in their body as compared to patients to the extreme right, which are more yellow, and yellow means a lot of fat in the muscles, okay? So this type of analysis is fantastic, and Springbok can analyze individually 130 muscles, if I remember correctly the number. And this is a way to really characterize individual patients, and you see the heterogeneity can occur among different patients, and within one patient, left or right part of the body can be quite different. You have individuals that have a lot of yellow on the right leg, while they have still a lot of red on the left leg. And after this disturbing picture, but which is really very important for patients, and the global FSHD Foundation in Australia is managing that every patient in their national register has a picture with imaging showing its fat progression. Now, I want to present you an artist from the Netherlands who has created a kind of show with a rubber duck that is a giant duck. So I don't know if it's clear to you because the picture shows the guy really close to the duck, but look at the boats behind the duck and the buildings behind. So you will see that better here. Okay, so this is DUX4 in the harbor of Hong Kong. And you see the size of the duck as compared to the boats next to it. So DUX4 is a toxic protein. You have to get rid of it for therapy. What is the best way to get rid of an inflated duck? Well, it's you use a pin. And then what happens, with the pin you can neutralize the duck. And see what happened to that poor duck on the right in the harbor of Hong Kong. So that's what we want to do in your muscles, you guys. We, I mean all the researchers, clinicians, pharmaceutical companies trying to develop drugs against DUX4. So it's nice to want to kill DUX4, but how would you be able to say that you have been able to kill DUX4? DUX4 is only expressed in very few nuclei at a given time. It's very difficult to detect. So how will you know that patients are doing better? And so there are very interesting things ongoing for several years now, which are research on FSHD natural history. So this means, how does the disease evolve without any therapeutic agent? And these are the names: Resolve, Move, PETS for the kids. You have to be able to evaluate muscle function. And you know that when you go to the hospital, the medical doctors make you make all types of exercise and check how able you are to do them. And in addition, now there are a lot of investigation on asking patients what they feel, how they think their disease is evolving, what are the things they can still do in everyday life or not. And this is another way to evaluate muscle function. Now, researchers need biomarkers of disease progression. And most of them use muscle biopsies. This is no fun, this is painful, and moreover it's very difficult because DUX4 is being expressed in rare cells at random, so you never know exactly where will the surgeon take the biopsy. Will he pick up a spot where DUX4 is supposed to be expressed or not? And most of the time it's missing DUX4. And looking for biomarkers in the serum, and this was really highlighted in the meeting. It's very important because they're just on a blood sample. You can detect either a protein or an RNA, and that would be reflecting what is happening in the muscles, because you have the blood vessels going in the muscles. They will take what the muscle is secreting, and from a little tube with your blood sample, this protein coming from muscle, activated by DUX4, will be detected. Now, there's also the whole body MRI, as I just showed, and with a measure of fat content. And so there were two talks. So I've put the name of the first author, Friedman, in brackets because he could not make it to the meeting. And it was Sylvia Blemker from Springbok, the famous company showing fat progression in their muscles, who made the two presentations. And basically what these people are doing, I'm not going to go into details, is to try to find parameters that show fat evolution in the body of one patient. Remember, it's changing very much from one patient to the other. So in a given patient, fat evolution, can we have a way to predict his fat in 1 year from now? And if this tool is existing, then we give a drug to that patient, and we can hopefully see that fat is evolving less fast than what it was predicted from these studies. Okay? So now another search for a biomarker. So this is again my cell. And I show you something very strange that has been found rather recently, like 10 or 15 years ago, that the cell is making kind of bubbles. And these are called extracellular vesicles. So you see to the left part down of the slide, those little bubbles coming from the cell and taking a little part of the cell membrane and going out. And within those bubbles, you have things, molecules that were inside of the cell. So these little bubbles, these extracellular vesicles, contain RNAs, contain proteins that are typical of the cell from which they come. And this is now something that goes into the blood serum. And there were 2 presentations on that theme, trying to identify markers of FSHD inside of those bubbles. And so, unfortunately, the first person, Nizar Saad, could not make it. You know, it's becoming really difficult for a scientist who is not of American nationality, working in the United States, to get out of the country and especially come back, being afraid to be blocked at the border. And when you are married, you have children, you have a house, you have a job, and being thrown in jail when you come back to your country is something that Nizar did not want to dare. Okay, so but the data are there. He identified 2 proteins which are very interesting in those extracellular vesicles. And then the other study was from Rome, Elvira Ragozzino. She used the same strategy, but identified special RNAs that could be used as marker. So in extracellular vesicles, which are in a blood sample. Okay. Now, measure of disease progression, another story is from Julie Dumonceau, who is advisor to the association. And she is studying myostatin and was wondering whether it could not be used as a biomarker. So what is myostatin? Well, it's a protein that prevents excess muscle growth. So in Belgium, we are very proud of these animals, which we name Blanc Bleu Belge, so which have very large muscles. They are named double muscle. They have a mutation that has killed their myostatin gene. So myostatin is a brake that prevents muscle from becoming too big. If you break the brake, then the muscles become big. And the problem for those animals is that when the cows are pregnant, they cannot deliver their baby naturally. You need a vet to make a C-section because the baby, the calf, is too big. Its muscles are way too big. Okay? So this is making you all fantasize, wow, if I could have anti-myostatin, I would have big muscles. And indeed, it was thought that for many neuromuscular disorders, it could be a solution. And at least 5 different companies have developed agents that could block myostatin. But unfortunately, as you see written in white in this slide, failure of many clinical trials in neuromuscular disorders with this anti-myostatin. And guess who discovered why? Julie Dumonceau. So what she published, she said, okay, these companies are giving drugs that block myostatin, but have they checked that there was myostatin in the bloodstream of those patients? Well, the companies had not. So Julie very simply took blood sample from people with various neuromuscular disorders and assayed for myostatin, and there was none of it. So the drug could not act because its target was not there anymore. And so atrophic muscles do not produce myostatin. I mean, your body is very nicely balanced. If you have muscles that are starting to become too big, they will produce myostatin to slow their growth, to keep up good balance. But if your muscles are becoming too thin, they will not produce myostatin. They need to grow bigger, so they don't need a break to becoming bigger. So there is this balance that had been overseen by people. And then she reasoned further, okay, problem with the myostatin, anti-myostatin strategy, but then the amount of myostatin in the blood in patients with FSHD could reflect the amount of muscle that those people have. The more muscle they have, the more myostatin they will produce, and you detect that in the blood sample. So she found that myostatin decrease in the serum could be considered as a very good biomarker of disease severity in FSHD. Now, in contrast to what I just told you, there was a presentation by Adam Vogel from a company named Scholar Rock, and he is using DUX4 mouse model, and he is giving anti-myostatin agent. And he said there was some increase in muscle mass, and there was increase in muscle strength. So does it make sense with what Julie had thought, that atrophic muscles do not produce myostatin? So atrophic muscles in mouse expressing DUX4 should be the same. But in fact, because DUX4 expression is only in a very small number of cells at any given time, the reasoning was that there were still, let's say, 90% of the muscles that could produce myostatin in patients with FSHD. And so this myostatin could be targeted by anti-myostatin agent, and same thing in the mouse. And in the future, the best strategy will be to use two drugs. One drug that is a DUX4 inhibitor, so the nasty duck is killed, it's not destroying muscles anymore, and the anti-myostatin, because the muscles feeling better are now producing myostatin, and there's a target for this strategy, and that will help muscles to grow bigger faster. Okay? So I will speak a little bit now about ways to change the structure of chromatin, so to make chromatin tighter, close to prevent DUX4 expression. So in FSHD2, most of patients have a mutation in the SMUSH1 gene, and that is killing the protein. And so you have presentation of this structure of protein. It's making a clamp, so two proteins associate in a dimer. They make a clamp, and they can bind to DNA. And they can pile up over each other to oblige DNA to make large loops and to block those loops with very close chromatin in them. So this is something very interesting, and it's the first time in a meeting that we have a researcher from the University of Singapore coming with a very smart idea. I will try to make a small gene coding for a mini smooshed one that will have the ability to bind DNA and force it to make a tight chromatin to block DUX4 expression. So it will have to be tested. Okay, I don't know how I'm doing with time. Not good. Not good. That's always my problem. I can stop any time because...
it depends. Either you want time for Q&A or we save the question for later and you finish until 11.
Yeah. Okay, so what do you prefer?
Q&A or highlights?
Listen to Alexandra.
Highlights?
Okay.
Yes.
Okay, thank you. I love to speak, as you have probably noticed. So I have to speak now about what is happening in muscle regeneration. So not only do you have muscle cells that obviously have to divide and to fuse to make the muscle fibers that were destroyed by DUX4, but you have help from other cell types. And among these are the FAPs, fibroadipogenic precursors. So these cells are kind of stem cells that, according to from where the wind is blowing, can decide to become fat cells or to become fibroblasts that can make fibrosis, that can make the scars. And this is a problem that you have in FSHD. You have too much fibrosis, so these are the extracellular matrix produced by the fibroblasts that come from the FAPs, or you have adipocytes, which are the cells that can fill up with fat. And another cell type that is playing a key role in muscle regeneration are macrophages. And you have two types of macrophages. You have the ones named M1, which are acting first. So when a muscle has been injured or has been partly killed by DUX4, you have the thank you. You have the pro-inflammatory macrophages. So these are the cleaning team. They remove all the leftover, all the debris of the dead cells. And when they have finished their work, come the second wave, the M2, which are anti-inflammatory, and these are helping muscle cells to proliferate and regenerate. So these are examples of cell types that you find are necessary to help to regenerate muscle. And so one of the very interesting talk was from Beatrice Biferali in the group of Davide Gabellini in Milano. It was understanding and treating inflammation in FSHD. So I make a quick slide with a summary of what they found. So what I'm writing here is the different steps of their reasoning, but most of the data were obtained from other researchers in the world, especially Stephen Tapscott's group in Seattle for the first line. So DUX4, among the different target genes, among the different music instruments it's activating in muscle where they should not be activated, are some that will produce special RNAs which are 2 strands. So you know DNA is 2 strands, RNA is 1 strand, but here we have RNA with 2 strands. And this is something very strange in the cell that is reminding of infection by some viruses. And you know the COVID virus is a virus with RNA. And so the cell has a whole way to fight viral infection. And this is by producing a signal, a protein signal that goes into the bloodstream and that will give the alarm to all cells in the area, all cells of the immune system. It is named interferon gamma. And so it's known in FSHD, you have increased interferon gamma. And when interferon gamma is there, it will activate cells to produce interleukin-6. And interleukin-6 is another type of signal, and which is also typical of inflammation. So interferon gamma, interleukin-6 are typical of inflammation and can be measured in blood of patients as being increased. And by the way, there is a clinical trial ongoing at the University of Nice with Professor Sabrina Sacconi with anti-interleukin-6. And we don't know the result because the trial is ongoing, but that's the idea, to suppress this alarm signal, this inflammation signal. And so because of this cascade, so double-stranded RNA, the cell thinks it is invaded by a virus, it reacts, makes interferon, makes interleukin-6, and inflammation, fibrosis, fat deposition. Because inflammation is telling the FAPs, the fibroadipogenic precursors, make fibroblasts, and that will make fibrosis, make adipocytes, and that will make fat deposition. So we are in trouble. And what is clever is that Beatrice found the idea of blocking interferon gamma, and it was a much important improvement in the mouse model on which she was working, the DUX4 mouse. Okay? So there are new mechanisms of DUX4 toxicity which are discovered all the time. This is really a very nasty duck. And so very quickly, so the group of Adam Bittel, who also from the United States did not make it to this meeting, is something named ferroptotic stress. So discovered that in cells from patients there is an excess of iron. And iron favors oxidative stress, and you know oxidative stress is a major issue in FSHD. And so trying to get rid of this excess iron. And then the group also in Milano, in Davide Gabellini's group, they found protein that can activate DUX4 production, and they are developing with a pharmaceutical company agents, so small molecules that can be injected in the blood in their mouse model that block this activator of DUX4 to decrease the amount of DUX4 present in the cell. Okay, so I will finish now by the hot stuff. There are now about 5 different companies which are using a strategy to destroy the DUX4 messenger RNA. And this strategy is a small molecule, a small RNA, which is named small inhibitory RNA or microRNA. And when you put this RNA, which has been designed to stick onto the DUX4 messenger RNA, it will push the cell to destroy the complex of those 2 RNAs. Again, it's the same story. You make a double-stranded RNA, the cell doesn't like that at all, and will destroy it, thinking it's a viral infection. Okay? And so what happens, if you destroy the DUX4 RNA, you prevent production of the protein. Now, this is a slide from Avidity Bioscience, and you see in purple the siRNA that it has been using to target DUX4. And in order to get it to enter into the muscle cells, it has been coupled to an antibody, and this antibody was raised against a protein that is present on the surface of the muscle cell. And this protein is named transferrin receptor 1. Interestingly, it's transporting iron into the cell, by the way. Okay, so that's the trick. You have the drug, and it is hooked onto a targeting agent, as if you were putting an address label on your postcard. And that will send it to muscle in priority. So the gray things are presenting muscle fibers. The little squares upon them are the target protein onto which the antibody in blue can bind. And so in this way, you can inject in the bloodstream the drug, and with its address label, it will go to the muscle, bind to this protein on the muscle surface, and enter into the fiber. And so this is something that is very interesting, and that is apparently working pretty well. A clinical trial of 1 year with Avidity is giving a lot of hope that things are really improving in patients, and they are starting now their phase 3 clinical trial to have extended work. And other companies are using similar strategy, and Dyne company is a little behind. It has not started the clinical trial yet, but it presented data that if you let DUX4 being expressed in a mouse in order to get the mouse muscles in a very bad shape, and only after that you give the drug, you can recover really muscle. So it's really a good hope. So I will, so one last thing is in Roma, Lorena Di Pietro is studying the different cell types that you have in the muscle. So you have the muscle cells, you have the macrophages, you have the FAPs, you have the fibroblasts, you have the adipocytes, and still other ones from the immune system, and she's trying to study that. Now, you've been all singing for Happy Birthday to Nicole. I don't recommend you to sing for Happy Birthday to DUX4. It's a little more than 25 years that we discovered DUX4 in my lab, and we wish him a catastrophic birthday. Thank you for your attention.